So B cells are coded in their B cell receptor, which is an antibody. And if multiple antibodies on the surface of the B cell are binding with an antigen, because maybe it's a repetitive antigen, right? Maybe it's a polysaccharide with a lot of repeating subunits, right? A lot of the sugars repeat. Like lipopolysaccharide, which are part of gram-positive or gram-negative. Gram-negatives, right? That's a repetitive molecule. That molecule will very easily activate a B cell on its own, lipopolysaccharide. And that B cell will produce IgM, though. That's it. That's a T-independent antigen. So lipopolysaccharides, polysaccharides, right? These repetitive molecules can stimulate the B cell without T cell help. But it's going to be stuck with IgM. Because remember, that's the mega big one. It's pretty much going to stay in the bloodstream. It doesn't cross the placenta. It can activate complement and things like that, right? But it's limited in its action. We don't want to really make other antibodies like IgG and IgA and IgE and G without confirmation from another cell, like a helper T cell, that yeah, we really need to step up our game, right? And have these antibodies that do more, like IgG that can cross the placenta, and IgA that's secreted into everything that we secrete. Make sense? Okay. So we've been talking about this, right? The importance of those abilities, those markers that allow us to present antigen. So on all our nucleated cells, it's MHC class. Well, that's our number one protection, right? MHC class 1. Then class 2 is our more specialized presentation of antigen, and this is by our antigen presenting cells. Who's classified as an antigen presenting cell? Nope. They have antigen presented to them by who? Who takes an antigen, processes it, and displays it on their surface in MHC class 2? Dendritic cells are our main antigen presenting cells. Yep, they're really important for activating both helper T cells and cytotoxic T cells because they have MHC class 2 and 1. But they and 2 other cells are the only ones that have MHC class 2. So who else can gobble up antigens? Macrophages. And then a B cell, right, has to take into the antigen, and it also has to present it if it needs help from the helper T cell. So your antigen presenting cells are your macrophages, your dendritic cells, and your B cells. Yep, those are your antigen presenting cells. So remember, the T cells and their CD4 markers, their CD markers determine which one of those MHCs they're going to talk to, right? So helper T cells have CD4, which allows them to help out, right? And who are they going to help out? Who's presenting antigen that needs help from a helper T cell? Yep, class 2, and that's found on macrophages, dendritic cells, and B cells. CD8, on the other hand, those are cytotoxic T cells, right? They're going to eat, they're going to destroy anything showing foreign proteins. So which class of MHC is that do they interact with? 1. And where do you find that? Where is MHC class 1? What cells have MHC class 1? Dendritics have it. Macrophages have it. All nucleated cells in your body have MHC class 1. Because any one of them could get infected, say, by a virus, right? Or a bacteria. They have to have ability to present this information. And unfortunately for them, is if they're presenting the bad guy and a cytotoxic T cell recognizes it, they're going to kill them, right? But we have to have a way of detecting that. And that's what MHC class 1 helps us do. So how did we learn about MHCs? Well, it stands for Major Histocompatibility Complex. What were we trying to do? When do we have to match these things? So, my heart is failing. I need a new one. Right? My cells have MHC class 1, right? So they can present to my cytotoxic T cells when they're in trouble. What happens if we stick a heart in me, different MHC? My immune system's going to attack it because it can't communicate with it or doesn't recognize it. So people who get organ transplants have to be on immunosuppressant drugs for the rest of their life, with one exception. Do any of you guys have an identical twin? Yeah, identical though. Because identical means just that. Right down to your DNA, you're identical. So that's the only other person out there that would potentially you be able to get an organ from, and your immune system would be like, oh, well, this is mine. Right? It's exactly the same. Fraternal means you just shared the womb together. Is it a sister or a brother? Sister. Yeah. But you could have a brother, right? Because you're a whole different egg and whole different sperm. You just happen to be in the same place at the same time. Right? Identical twins is the only one that would help you out. You wouldn't have to be on immunosuppressant drugs for the rest of your life. Siblings, relatives are more likely to be close matches, right? Because genetically you should be similar if truly you are genetically linked in that way, right? You can have stepsisters, right? That's by marriage. Half sisters, right? That means you share one parent, not both. So that's when they found out about these. And then they're like, oh, okay, now we understand, right? Cells are using these markers, right, to communicate. So it makes sense when these don't match that the body can't communicate and attacks these organs, right, that we try to give people. So they've tried to engineer, actually in pigs. Pigs actually are really closely genetically close to us, believe it or not. They tried to make it so that they could genetically engineer, like if someone needed a heart, to like get the pig to be the same as that person. Last I heard, they weren't so successful at that kind of stuff, though. Right, but if you watch sci-fi movies and stuff like that, they talk about cloning people and having clones, so that if you need excess body parts, then you could just get it from your clone, who would be genetically identical to you, so you wouldn't have any issues. But yeah, people who've had, anyone know anyone who's had organ transplant? Nope. So as I said, immunosuppressant drugs for the rest of their life. They don't go to crowded festivals, right? Because their immune system is purposely being weakened so that organ will survive and they will survive. So that puts them at greater risk for infection. So they avoid situations that would put them at greater risk, like festivals, and they certainly wouldn't work at a daycare. Those things are little germ magnets walking around, right? So to be careful of those types of things. But again, it's all about this communication, right, that needs to happen between the immune system and our body to help defend us. So let's see. I'm trying to think. There's a lot of the repetitive of the same stuff. Oh, natural killer cells. So there's two important outcomes. So these are also lymphocytes, right? But these guys are not antigen specific. They don't have antigen receptors on their surface. They don't have, receptors like T cells do and B cells do. But as their name implies, they kill, right? And one of theirs is aided by antibodies. And it's a particular outcome that can happen with IgG antibodies, to be specific. If an IgG antibody binds to a cell because it's recognizing an antigen that cell is presenting, the natural killer cell can come along. It has a receptor for that FC region, that region of the antibody that our immune system interacts with. It has a receptor for that, for IgG. When it binds to that, it's going to kill this cell that's coated with antibodies. So it's killing that cell because the antibody is recognizing it. So this process of an antibody and a natural killer cell killing a cell is referred to as antibody dependent, right? You've got to have the antibody for this to happen. Cellular, because it's a cell. Cytotoxicity, because again, we're going to cause the natural killer cell is going to cause the death of that cell that's coated in antibodies. It's right here. Antibody-dependent cellular cytotoxicity. Abbreviated ADCC. Right, the FC region is this portion of the antibody that sticks out, right? And so some of our cells have receptors for that part of the antibody, and they're going to grab onto it. And that's how he's able to recognize what's going on. So the other thing is that some viruses, when they get inside of our cells, they shut down antigen presentation by MHC class 1. So notice this cell is presenting, and all the time they're even presenting their own proteins. But of course, our T cells shouldn't recognize our own proteins, right? Because we've eliminated those in the thymus if they recognize self. So here, the virus gets in, and it actually shuts down antigen presentation by MHC class 1. So notice there's no MHC class 1 on the surface of this cell. When the natural killer cell comes along, and he doesn't see any MHC class 1, that's a sign that there's something wrong with that cell, because it's not doing what it's normally supposed to do, right? So, if you go somewhere and they ask to see your ID and you don't show it to them, well, one, if you weren't trying to get in somewhere, they're not probably going to let you in, right? And if you are acting strange and you're not showing your ID, you're not acting normal, then they're going to be aggressive towards you, right? Same thing here. If you're not showing your identification, you're not showing MHC class 1 like you're supposed to, then you're probably not doing what you're supposed to, and they're going to kill you, right? So some viruses can avoid detection, at least for a little while, by not having us present. But the natural killer cell is our backup to that. If a cell isn't presenting MHC class 1, they're going to eliminate it. Make sense? So these guys, they use components of the immune system, but they don't specifically determine, it's the HIV virus or the influenza virus that's inside that cell. No, they're killing that cell because that cell is not doing what it's supposed to do. It's not presenting antigen. Where in this case, right, if the virus is causing it to present proteins associated with the influenza virus because it's taken over that cell, if we've got antibodies against that protein, those antibodies will bind, and that will help facilitate the natural killer cell to kill that virus-infected cell.
Is there an atypical one?
No. It would just be not presenting MHC class 1. Okay, so this guy is really good, right? You remember this from the very first website I sent you guys to? Which, don't forget to finish that discussion board, y'all. The discussion board where you got to share something for the web, your own web search, right? And reply to 2 of your classmates. I know we got kind of sidetracked off of it, but try and get that done for Tuesday. It'll probably help you, right? Because people posted some cool stuff. Okay, so they're showing this cute little guy, right? So he's a dendritic cell. He's engulfed something. He's presenting. What are they not showing you here? This is the antigen, but what is he using to present that antigen? He's presenting in MHC class 2. And that's because he's talking to little helper T cell there. And helper T cell can talk to this dendritic cell because he has CD4, right? And he recognizes it, so he's all happy. He becomes activated, right? And he proliferates, right? That's antigen presentation. So now he can go help his buddies. So here, right, he's helping out Mr. B cell that recognized a specific pathogen here, this bacteria. He internalized it and he's presenting it. But you don't see that right here, right? They haven't drawn in the details there. The details we know that we would draw in, right, is that this helper T cell has CD4. This B cell is presenting the antigen over here to the T cell by MHC class 2, right? They're communicating, they're recognizing, so this guy becomes activated, he proliferates, he turns into what cells? Plasma cells and memory cells, right? So plasma cells and memory cells. And the plasma cells are so cute because they literally spit out, right? Secrete antibodies. And one of the potentially helpful outcomes is these antibodies will specifically bind to that bacteria, right? And right down to his cute little crown he's wearing, right? That epitope, that little piece even of that whole antigen. This very small molecular interaction of this antibody with this pathogen, with this bacteria. And then, of course, we have cells that can recognize when stuff is coated in antibodies, right? Phagocytic cells like good old macrophage. He has a receptor, again, an Fc receptor for some of the antibodies, like IgG. And he could bind to it and destroy him. And then later on, if the bad guy comes back, right, those memory cells we produced here will probably be the ones getting activated, and we start this all over again, right? But definitely much more aggressively here the second time, right? We're going to produce even more antibodies even faster. So maybe or maybe not, right? I did, for those videos that you guys watched, I actually drew on these whiteboards in here, right? And one of my students videotaped me. I used to say to my students in the past, draw it out. And they looked at me like I was a crazy person. So I was like, well, you have to teach them, Erica. You have to draw yourself first. So though not my best thing, right, drawing, and it doesn't have to be beautiful. It doesn't have to be adorable like this, right? But the actual act of, and that's why I asked you guys to take notes, but if you didn't draw like I did, you might want to go back and actually physically draw. I'm not going to look at them. They're for you, right? So it can be messy. It doesn't have to be beautiful. It doesn't have to be cartoony. But the actual process of trying to draw out what's happening helps a lot of us in that learning process, right? So that's why I say draw it out. Don't just write it out, but drawing it out. Even just flow charts with words sometimes. Again, you don't even have to do the cute little circles. Flow charts with words can be helpful as well, right? So that brings me to the other thing I love to do, because it makes sense for me. I do want to save, because I need to save that slide. I'm not even going to close this just yet, except I just did. All right, I'll open it back up later. So now we're going to talk about disorders. And so there's a really helpful table, right, for disorders. I can't remember if I posted it blank for you guys or not. Let's see. Giardia. I put that as the 20th. All right, we'll talk about this Tuesday. Don't stress about that if you haven't done it yet. But eventually you do have to get test 4 done. Okay, I'd rather you guys study over the weekend for test 3. So in other words, I'm willing to move that date. Because I don't even know when that is. What is that, Monday? I don't even know. Yeah. You just got to get it done before I have to submit grades. 27th. Or the 28th. One of those two. Let me put it this way. I need to be done by the 28th. So you need to be done by midnight the 27th. Anything you want to go towards your final grade.
So they try to get, like, everything will be in by the 27th and 28th for teachers.
Okay. So let's see. Links. No, that's not what I want. Back. I'm looking for this. All right, so, yep. So there's the table in a blank form, right? So you could print it out and try and fill it in yourself. This one's really helpful. Like, I highly, highly recommend you do this for disorders. So, which is what I want to open up. I don't know why I have two things open. So disorders are when things go awry, right? And this is our last topic. So within disorders, we talk about hypersensitivities. There are 4 different hypersensitivities. Although I'll say type 1, 2, 3, and 4, I'll give you guys the full names of them on the exam. I don't want you memorizing what 1, 2, 3, and 4 are, because you're not going to remember it later on in life, and it's going to not help you. Autoimmune, I'm not going to go over. You can pretty much go over yourself. And as the name implies, it means the immune system is attacking self, right? Most of us are aware of that problem. And immunodeficiency, right? Most of us are aware of the fact that HIV can cause your immune system to be deficient, and that's where it gets the name acquired immunodeficiency syndrome, the acronym AIDS, right? So people's immune systems, they were acquiring something that was causing it to be deficient, right? And there was this list of symptoms. They didn't know what was causing it for the longest time. Come to find out, there's a virus that can cause your immune system to be deficient because it obliterates your helper T cells. And we've already learned how important those guys are, right? Kind of don't really want to be without them, right? Otherwise, your immune system becomes deficient. But with the special drugs and the things that we've done nowadays, people are living with HIV and not progressing to the AIDS status as quickly as they used to in the past. So they're no longer synonymous, although people somewhat use these two terms synonymously, and they are not, right? HIV and AIDS are two separate situation conditions. And that's an important distinction that unfortunately gets blurred. So there are other things that you could acquire that would cause your immune system to be deficient. And so we could technically say you had AIDS, right, and not actually have HIV. Okay. Although AIDS, that acronym has pretty much become synonymous with HIV. But understand it stands for acquired immunodeficiency syndrome, right? They didn't know what was causing it at one time. Malnutrition can cause your immune system to be deficient. As we age, our immune system wanes. If you had a transplant, right, we've purposely made you deficient by giving you immunosuppressive drugs. These are all acquired ways, right, as opposed to primary immunodeficiencies where you're born deficient, right? So where you genetically cannot do what you should do to have a truly functioning immune system. And there's different degrees in that and different abnormalities. But most people have heard the other acronym, SCID. You guys heard SCID before? Stands for Severe Combined Immunodeficiency. These are individuals who are born with not being able to make functional B cells and T cells. So basically no adaptive immune response. They just have the innate immune response. This is basically the bubble boy, right? So it tells you the likelihood of having this defect is so many in so many live births. Because many people who have this defect die in utero during development. They're not even born. If they are born, then if it's not diagnosed soon enough, infection will kill them very early on in life. And yeah, they basically have to live in almost a sterile environment as best as possible to stay alive. Because as you know, we're surrounded by microbes continuously, all the time. Does that make sense to you guys? Then there's different ones, where people don't make IgA, right? So they're more susceptible to respiratory and digestive infections, right? Because they don't have that protection in their mucus of having IgA, right? Others that don't produce a lot of antibodies. I had a student a couple of semesters back, her daughter had aglobulinemia where she produced almost no antibodies. And so she'd have to go for regular transfusions of antibodies from other people. And they're not kidding when they say you could save up to 5 lives when you donate blood, because there's so much more than just red blood cells they get from your blood. They can get plasma, platelets, antibodies. So there's so much more that can be used other than just the red blood cells. And they do separate it out, and they take your red blood cells out from your plasma because they don't want your antibodies causing reactions for other people. When you're given red blood cells, it's what they call packed cells. It's mostly red blood cells and fluid. They've removed the person who donated its antibodies, so you don't have any cross-reactivity. And then they can purify out those antibodies and use them for anti-serums against tetanus and diphtheria and things like that. If you're hyperimmune, if you have a lot of those antibodies. So we're going to really dive into the sensitivities and hypersensitivities, the 4 different types now. And like I said, if you fill out that table, right, that would really help you. And so that this is the table. I opened it, right? No? I just thought I did. But I did see it was posted for you guys as a blank table, right? So as we look at each one of these, right, this is type 1, type 2, 3, and 4. I didn't even put the different numbers, right? I put the names. So in the name itself, it tells you a lot, right? This one is immediate, right? This happens right away. And the reason why it happens is because of IgEs, right? So this is what most people who experience allergic reactions, right, that can be literally immediate, are dealing with an IgE problem. Cytotoxic, as it implies, cells are being killed, right? Cyto, cells, toxic, kill. Immune complex mediated, this is because of immune complexes. And what immune complexes are, antigen and antibodies. Delay cell mediated, all of these 3 actually are mediated by antibodies. This one is not, it's cell mediated. So you're dealing with T cells and macrophages. And because it's cells, it's a little bit slower, so it's delayed. For each one of these, you should know the type of antigen, and especially for these 3, where it involves an antibody, what antibody. Well, this one, it's nice, it tells you in the name, right? IgE. What other cells might be involved, what mediators, how long does it take on average for the reaction to happen? And is there a skin reaction? And if so, what is it like? So that table can be really helpful in digesting all the information for these 3 disorders. So type 1, as I said, the name tells you a lot. It's immediate, it's IgE mediated. So, trying to think, there's the types. So same progression. I'm trying to think where I want to go with this. Yeah, I want to go right here. So same thing that happens whenever we're exposed to something. The problem with this, why we call it a hypersensitivity, an overreaction, an oversensitive, is that B cells are being stimulated by antigens that wouldn't actually harm you. Ragweed pollen will not grow in your body. It will not harm you. But for some of us like myself, when we inhale it, our B cells recognize it. The helper T cells help out and go, yeah, that's bad, right? And convince this guy to proliferate and to class switch to IgEs because of where this is happening. And all this is programmed, remember, in our DNA. So guess what? I have my mom to thank for this. I inherited this from her. And my poor son inherited it from me. And why do we keep passing it down? Because, you don't die from things like allergies to ragweed, right? But peanut allergies, on the other hand, people used to die from that, y'all. And that's why we see more and more of it, because we know about it, right? So people aren't dying from that peanut butter and jelly sandwich in the first grade. Instead, they're growing up avoiding the peanut butter because they learn early on about the allergy. They don't ingest it again because each time, remember, each time we get exposed to something, the response is bigger and faster. So that first one may have sensitized, started this whole thing off, and you don't even know it. Then the next time you eat that peanut butter sandwich, right, you respond. Then the next time you respond even more aggressively. So that if you're stupid yet again, you may potentially die. So you don't grow up and have babies who have peanut allergies because you died. You don't pass it on. Right? That's one of the reasons why you see more allergies now than we did in the past. Because we know about them, we can avoid dying from them. With the ones that especially are ingested. Right? So they carry around EpiPens, epinephrine, right? Constrict the blood vessels, increase heart rate, get you to the hospital in time to support you while we get you through this crisis, right? So, first time you become sensitized, you produce these damn IgEs. Unfortunately, these class of antibodies bind to mast cells and basophils, right? The effector cells that release histamine. These suckers, these IgEs, stay attached to those mast cells and basophils for weeks at a time, right? So once you've got them, once they've coated these cells, the second, I kid you not, for some of us, the second you inhale one of those allergens, it binds to these antibodies already on the mast cell. It degranulates, release the histamine, congestion that fast. Literally within seconds. I was at Gustav, one of the hurricanes. I came back, walked into the building, and immediately could not breathe. Immediately. From all the allergens that got stirred up from the hurricane. So this reaction can happen, say, in your nose, and even for some people, their eyes will get affected, right? This is commonly referred to as hay fever. The good news is it's really freaking uncomfortable, but you won't usually die from it, right? Hives, again, this is the reactions happening in your skin. And you see this wheel and flare. And what you're seeing here is actually somebody's allergy test. And I had this done. I couldn't wear my watch for 2 weeks and had to take steroids after they injected thistle into my wrist. Okay? It was pretty freaking scary. Wasn't allergic to any animal stuff. That was on this arm. I was threatening to cut this arm off during my allergy test because, I wanted to be a veterinarian. I had lots of animals. I still have lots of animals. I didn't want to have to get rid of them. No, it's everything outside, right? This entire arm was like, if I had known I was going to be a teacher, I would have took a picture of my horrific arm because it's nothing compared to this picture, right? So, Some people will get this if they come in contact with something, right? And of course, I've had that happen to me. Some people will ingest it and get hives. Because what happens is, think about it, it goes into your bloodstream and it goes throughout your body. So your skin is part of your body. So guess where the first place it usually shows up if it's something you ingested and you're having an allergic reaction to it and you're having a skin reaction? Which skin gets the blood first from something you ate? Your stomach, right? When the hives start at the stomach and go outward, it's usually something you've ingested, right, that you're allergic to. It'll spread up the chest and the arms. If it's somewhere else, it's probably maybe a contact problem. Right? And if you're allergic like me, I had a really nice Vera Bradley cloth purse last summer, and something got on the handle, and I got it on my arms. Again, steroid cream for weeks to get rid of the bumps. I still got some. It didn't completely go away. Not fun. So then you buy things that you can wipe down on a regular basis. So you don't have this, right? Some people, it happens in their lungs, right? Something they inhale, and that's a little bit different. That's not histamine, it's other mediators that release. That's why they take different types of drugs than people who have hay fever-like allergies that usually take antihistamines. But same thing, right? You're recognizing something foreign, you're responding as if it was a big, bad, ugly thing. Unfortunately, it's in your lungs, right? And your bronchioles tend to constrict, which decreases airflow to the lungs. But unfortunately, you kind of need to breathe to live. And it's quite terrifying, and I'm so glad I don't have that. I have a friend that does, in which he would have an attack. I would have an attack too, I think. Right? It's pretty terrifying to watch someone who can't breathe. So that one can, unfortunately, be life-threatening, depending on the level, right, of constriction that happens. And they have to carry relief inhalers. But the most terrifying is systemic, right? When something that you're allergic to gets into the bloodstream, right? And you have widespread inflammation, so much that you can go into shock because your blood vessels are dilating, right? And so therefore you're not getting enough blood flow everywhere in your body. And you can go into shock for multiple reasons, right? Excessive loss of blood, right? In this case. So a lot of times when you're in shock, they're going to try and give you fluids to get the blood volume up while they're trying to figure out why you're in shock, right? And you can even be in emotional shock, right? I was in a car accident once and I wasn't making sense. And the paramedic's like, oh, don't worry, she's just in shock. But it was emotional shock, not any injury shock, thank goodness, right? My system was flooded with hormones that were screwing with the system. It passed over time, right? But in the case of anaphylactic shock, right, we need to address the problem, right, so that you don't die. There are different therapies out there, and I've been through this therapy where they made a special cocktail just for me of what I was allergic to, and they would inject it into my arm, very low concentrations, and they would slowly increase it. I wasn't able to completely go through the therapy because I moved cross-country during the time that I was going through this. But I can say that I don't think I'm allergic to thistle anymore, and thank goodness that was the one that was really bad. Because I've been desensitized, my system switched over to IgG instead of IgE. And that was by very simply changing the way in which that antibody, the way that antigen was introduced into my body. So instead of inhaling it like I normally do, right, they injected it into me. So that hopefully it got exposed to B cells that were programmed to produce IgG. So now my IgG grabs onto that allergen before it gets to the mast cells, which are coated in IgE. And because it's been so long since I've been exposed, it hasn't been able to go that route, I don't have any more IgEs in my system. But I still have those memory cells. So if it was to get through, right, and the IgGs didn't get it, then I would have a reaction. But this is called desensitization, and a lot of people go through this, especially if they're really allergic to some allergens. But this therapy itself could cause you to be sick, right, if it's not done properly. This is a therapy, I'm not sure to the extent that it may or may not still be used, so I skip over this one now. I haven't had a chance to research it, so you guys don't have to worry about it. So the next is cytotoxic, and we really only found out about this when we started doing blood transfusions and also with hemolytic disease of the newborn. So this is when you produce antibodies against red blood cells. Two things can happen. Those antibodies can activate complement, and remember that MAC complex can destroy the cells that the antibody is coating. Or it could be IgGs, that antibody is coating the red blood cell, and then the natural killer cell comes along and attaches to that IgG and destroys that cell. Remember, that's antibody-dependent cellular toxicity. Both of those can happen with a transfusion reaction. As well as both of them can happen with hemolytic disease of the newborn. Because both IgG and IgM can fix complement. But only IgG can do antibody-dependent cellular cytotoxicity. So it depends on which antibody is causing the problem. If it's IgM, only complement fixation is happening. If it's IgG, it could be complement fixation and antibody-dependent cellular cytotoxicity by natural killer cells.
Well, is it IgM and complement fixation?
IgM is complement fixation only, not antibody-dependent cytotoxicity, because the natural killer cells needs IgG. So if you're having a transfusion reaction because of the major blood group antigens that we talk about, ABO, in the ABO blood group, then it's probably IgM, and so therefore it's just complement fixation happening and lysis of the red blood cells by the MAC complex. So what happens with this, uniquely enough though, remember I said how there's some pathogens out there that are kind of similar to us? You're naturally exposed to some bacteria that have antigens on them that are similar to the ABO antigens. So if you have O blood type like me, you've been exposed to bacteria out there that have antigens like the A and B antigens. And because I don't have A or B antigen on the surface of my red blood cells, those T cells were not eliminated in my thymus. So they made IgM against both the A and B antigens that would be found on our red blood cells. So if some idiot gave me either A or B, or even worse, AB blood type blood, I would have a serious reaction where my IgMs would attack those red blood cells. Although I've never had a transfusion before, right, so I've never been exposed to blood before, I would still probably have a reaction because of natural exposure to similar antigens during my lifetime. That's what we found out has happened with the ABO blood group. And it makes sense, right, that these pathogens have similar antigens, because for some people, right, especially for somebody who's, say, A-type blood, if that pathogen has the antigen that's similar to the A antigen, are you going to attack it? No, because you would have deleted those T cells and B cells because it recognizes a self-antigen. Your A antigen on the surface of your red blood cells. So that's bad news actually for people with AB blood type, right? So there are pathogens out there that have both the A and B antigens. You don't have antibodies against that. But it is beneficial if we want to give you blood, right? Because we can give you the A or the B antigens, and you're not going to have a reaction because you haven't produced antibodies against them because you have those antigens. So with blood typing, y'all, the easiest way to remember is just don't give anybody something they don't already have, right? You can't give them antigens they don't already have, right? So if you have B blood type, we can't give you A, right? Or in the case of me, O, which means 0, by the way, you can't give me A or B. We're going to have problems. Make sense? Now with the RH, that's a different ball of wax, right? This is where we're having fun with some Punnett squares. But I'm going to skip over that. The only moms that have to worry are moms that would make antibodies against this antigen, right? Because if you have what we call positive blood, we're talking about whether you have the RH antigen or not. I am RH positive. No worries. Because I have that antigen, am I going to make an antibody? No. But if you're RH negative, that means you don't have the antigen, right? So if you're exposed to positive blood, and for this one you do have to be exposed to this actual antigen, to positive blood, to make the antibodies. And in this case, the antibodies you make, unfortunately, are IgG. So this is why we end up with hemolytic disease of the newborn, because IgGs can do what? Boop. Cross the placenta. Because they're meant to help protect our baby, right? So if you're a negative mom and you get exposed to positive blood, you could make antibodies. And if you have a baby that's positive, because guess what? DNA comes from daddy too. He could give the positive gene even though you don't have it. Dad might have it, give it to the baby. So then the baby's positive, you're negative. Your antibodies cross the placenta and attack and kill those baby's red blood cells by antibody-dependent cytotoxicity by natural killer cells and complement fixation. So how do you get exposed to positive blood? As a woman, we have two things we have to worry about. Blood transfusion, and the second is pregnancy with a positive baby. If that placenta is damaged during pregnancy, that baby's positive blood could mix with yours, and you could produce antibodies against your baby. When that baby leaves, placenta leaves a hole about this big. I didn't keep my placenta, but my reproductive teacher when I was in college kept both of his grandbabies' placenta. They're this big. I mean, like you give birth twice. Once to the baby and once to the frickin' placenta. Right? The thing is enormous, right? Leaves that big of a hole inside of you. And baby's blood is in that placenta, right? It detaches. Some of that baby's blood can pass over into you. So for Rh negative moms, they actually give you antibodies against positive red blood cells to help stop you from producing your own antibodies, right? They try and they give you those antibodies to grab the baby's antibodies before your immune system recognizes and makes their own antibodies. It's called ROGAM. ROGAM.
How often does it happen?
Oh, this is very common, right? So Rh negative moms will tell you it's living hell, right? Especially if you are married to someone who's positive, right? And depending on how positive they are determines, like if they are homozygous, all your baby's going to be Rh positive, right? Because they're always going to be given that positive gene in the sperm to the baby. So Dr. LeBlanc will tell you about that because he must be homozygous because he has 3 kids. His wife is Rh negative. All of his kids are positive. And she had to go through shots throughout her pregnancy because, I don't know, at some point, she started making antibodies. And so they had to keep giving her antibodies to stop the antibodies to going across the placenta. Yeah, it's not fun. But, before we knew about this, babies literally would die from this.
But from an evolutionary know.
Right, yeah, I don't know why it would be beneficial to be negative, to tell you the honest truth. Like, it would probably be better that we would all being positive, yeah. Yeah, and it was actually, RH stands for rhesus monkeys. They discovered it in rhesus monkeys is how they figured out what was going on. So they have a similar thing that happens. But only negative moms, because again, only moms that don't already have this antigen are going to make those antibodies. So, and that's the other thing too, like miscarriage, depending on how far along in the pregnancy you went, that could cause a positive exposure as well. Right. So even though I knew I had O-type blood when I was pregnant with my son, I didn't know if I had positive or negative. But I wasn't too concerned. I'd never been pregnant before. I never had a blood transfusion. So I wasn't, the likelihood of me having been exposed to positive blood was extremely limited. Right. So I wasn't panicking. Right. But then when you go for your first, they do the blood test and you find out. And I found out I was positive. So then I didn't have anything to worry about. That makes sense? They also test you for a ton of sexually transmitted diseases too, because they don't want you to transmit it to your baby. And they don't have to tell you that they're doing it. All right. Type 3. This one is not covered very well in your current textbook. And I had wanted to print this out for you guys, but I didn't. But I did post it for you. So this right here, this type 3 document, definitely take a look at that. And it does a really good job of describing type 3 and what's going on with that. So type 3, this is immune complex mediated. So again, the immune system is exposed. It produces antibodies. But it produces, actually, I'm not even going to misspeak. I'm going to open the document. Oh, I can just do preview. It's only like a page or 2 pages. There we go. Right, so this is where, again, antibodies-wise, it's IgGs and IgMs, right? So these are the ones that would be found in your blood, right, and throughout your body. Usually they're rapidly removed, right, particularly if they're large. But this problem occurs when the Fc portion on the phagocytes binds to the Fc region of the antibodies, right? They engulf and destroy them. If there is slightly more antigen than antibody, right, smaller complexes will form. And these don't get cleared as easily as those big complexes, right, of antigen with antibody, right? This picture here, or where is it? They have it on this one. Yeah, so antigen and antibodies, right? That cross-linking that happens. If it's kind of small, it's not going to get gobbled up as easily, right? And there's more of them if they're smaller. And that you run into this problem when there's more antigen than antibody. And this is why I looked it up, so I would make sure I wouldn't say it in the wrong order. When you have a lot more antibodies, you'll get more complexing, right? Larger complexes. So these stick around. They can get lodged in blood vessels, right? And you can get neutrophils degranulating and complement activation happening in your blood vessels. So in really small blood vessels, like in the kidney, this can actually cause permanent damage to those nephrons that are needed for filtering your blood in the kidney. It can happen for some people throughout the body in the little tiny capillaries, and this really can impede blood flow to tissues. For some reason, too, immune complexes tend to settle in our joints. So you'll get inflammation in the joints due to these complexes settling out. Like I said, damage in the kidneys. And even in the skin, like you'll see a reaction sometimes in the skin because of these immune complexes not being cleared. I'm trying to think. There's one viral infection where you'll get that and you'll get a pretty characteristic rash. And I can't remember which one, and I don't want to misspeak. But what I find most interesting about this one too is the serum sickness that can sometimes happen. And again, this is when we inject something into you and you have antibodies. So I didn't close that, did I? I think so. The crazy snake people out there, right, that play with venomous snakes, blows my mind, but they do it. So arthritis is the name of the skin reaction in the skin, right, when you have that happening. Serum sickness is systemic, right, immune complexes, and this is due to passive immunity, where they've given you antibodies as a protection. So when someone gets bit by a snake, right, we give them anti-venom. That's antibodies against the venom. But sometimes, like if it's from another source, like I think they actually do this in pigs now, again because of that close relationship, so we don't get a reaction. But in the past, horses are hyperimmune to venomous snakes. And they'll actually get bit by a venomous snake and they'll be fine. It won't kill them. And they'll produce antibodies against it. So they would like purposely inject horses with venom and take their serum, take their blood, and fraction out their antibodies to make anti-serum. The problem with that is when they would inject it into people, the first time it would help you, but then some of those antibodies would be left behind, and your body would make antibodies against the horse antibodies. So you'd have anti-horse antibodies. So now if you are stupid and get bit by a snake again, and they got to give you anti-venom again, they can't really give it to you because you have antibodies against it. Right? You've made antibodies against it. So it can't really do its job because then you have your antibodies attacking the antibodies that are supposed to help you. So like I said, now they actually do it in pigs. And they don't have that cross-reactivity happening like they used to. But this can also happen, and I think they mention it here. They talk about the snake bites. Oh, diphtheria. So because of the potential cause of serum sickness, horse serum is no longer used, right? For passive immunity, also for diphtheria and tetanus. So diphtheria and tetanus produce toxins. And again, we would give you anti-sera, antibodies against those toxins. But if we gave you horse, then we couldn't give it to you again because then you produce antibodies against the horse. A lot of times, like I said, is people that are in the population that are hyperimmune, they're producing a lot of these antibodies in their blood, they'll take that antibody out of your blood sample that you donate. And they'll use that to make the antiserum. So that's human, right? And you don't have to worry about the cross-reactivity problem. And we can give it to you multiple times. Especially people who don't have functioning immune systems, right? If they're potentially exposed to these things, then we need to give them the anti-sera, that passive immunity, right? That we're passing on to them. But you have to keep giving it to them, right? Because they're not making these antibodies, someone else is. So they don't have memory B cells, right? Because they can't do it, usually. Does that make sense? All right, so it's kind of one of those freaky things where, again, where we're trying to help out, but we make the situation worse, right? Because the immune system goes, oh, I don't recognize that. I better make an antibody against it in case I see it again. So there is, by the way, a crazy snake guy who I think has purposely injected himself with small amounts of venom so that he's immune. And he sells his serum like for insane amounts of money because he has antibodies against like all the venomous snake venoms. And he works with venomous snakes. Yeah. He's cuckoo bananas. Cuckoo for Cocoa Puffs. Alright, so that's type 3. So make sure you read through that document. It actually does a really good job, I think, of describing. The last that doesn't involve antibodies is type 4, which is delayed cell-mediated. So this is where the cells of your immune system are recognizing something it's seen previously. And so actually the tuberculin test takes advantage of this, the TB test, where they inject you with proteins into your skin similar to the organism that causes tetanus. It's actually a cousin. It's Mycobacteria bovis. they give you. But it's close enough that if you have TB, if you have tuberculosis, your cells will recognize it, they'll infiltrate into that area, and you get that indentation, that skin reaction for this. That means that your cells are recognizing it, which could mean you have the infection, because you'll have the infection for a very long time before you're hacking up a lung, right? It's a slow progressing infection. The bacteria doesn't multiply that quickly in the lungs. So this is a nice early detection without having to do a chest x-ray. This is also why we don't immunize people in the United States against tuberculosis unless under certain circumstances, like if you work in a prison or you're in the military or something like that. Because once we vaccinate you, can we use the TB test? No, because we already told your immune system, hey, look at this. And actually, it's the same one we use for the TB test. It's bovis. I go back to here in bovis. So, if they've been vaccinated, their TB test is always going to be positive. I told you guys about my ex-husband in LSU, right? So you guys already know. And you already know that potentially someday, if you get a lot of TB tests, it might be like you've been vaccinated, right? And you might end up with a positive TB. Once it's positive, your cells are always going to react, right? You're always going to be positive. You'll have to get chest x-rays to make sure you don't have the infection. Make sense? So this can happen too with poison oak and other types of allergens. But the important thing to remember here, it's not an antibody response. It's a cellular response, right? The cells are recognizing that something foreign is aboard. And again, you have to have prior exposure. You have to sensitize these cells ahead of time. Ooh, we are just out of time, huh? And this takes, because it's cells that have got to move. They can't move as fast as antibodies, right? This can take 2 to 3 days. This is why they check the test several days later, not, Unlike when they do skin testing for allergens, y'all, IgE, I'm not kidding, immediate, right? Within seconds, minutes in that doctor's office, you see the flare and the puffiness. Yeah, there's no, oh, let's inject you when you come back 3 days later and let's see what happened. No, it happens right away. But TB, you have to wait, right? And so it'll look something like that. It might even have a bump to it, right, for a positive result. So as I said, for this, right, for comparing and contrasting, that table, right, really good. So fill it out, go through it, right? But again, it's a normal immunological reaction. The problem is it's responding to something that's not going to harm you, right? And so we don't actually need to destroy it. We don't need to destroy those red blood cells, right? We don't need to destroy pollen, right? It's not actually going to hurt you. But it is foreign, and your immune system's just doing what it normally does for foreign stuff. Attack. It's been a pleasure, guys. We'll see Tuesday night. All right. So remember, as I said too, if you want, we can go through this table. We can, if you guys have any questions, make sure you bring with you before the exam, right? We can powwow for a little while, or as long as you want, before we actually get on our devices and I put in that code.